We've all been there, right? With that extra cup of coffee in hand, trying to, like, convince ourselves it's fueling some incredible brain boost. But is it really the coffee or just wishful thinking? It's tough to tease apart what really causes what, right?
Hmm.
Especially with some things. Like, well, say we're wondering if having diabetes increases your risk of certain cancers. We can't exactly run an experiment and just, like, give people diabetes to find out.
It's certainly not going to get that past an ethics committee.
Exactly. That'd be a hard no from the IRB for sure. But that's what makes today's deep dive so interesting. We're getting into the world of Mendelian randomization, or MR for short. It's basically using our own genes as a stand-in for those cause and effect experiments we could never actually do.
You can almost think of it like this. At conception, we get this random shuffle of genes, almost like a deck of cards being dealt, right? And this hand we're dealt influences all sorts of things about us, from our height to our risk for certain diseases.
So if a specific gene variant makes you more likely to have, say, high cholesterol, and then later on you're also more likely to have a heart attack, that suggests high cholesterol might actually be a cause of heart disease.
Exactly. It's almost like our genes are running this giant experiment for us in the background.
I like our own personal research study written in our DNA.
That's a great way to put it, yeah. But I do have to add, it's not a perfect system. Mendelian randomization comes with some really strict assumptions. It's definitely not some kind of magic bullet for figuring out cause and effect. And it gets especially tricky when we're dealing with what researchers call binary exposures. That's really the core of what we're unpacking today.
All right, so let's break that down then. Binary exposure. That sounds kind of intimidating,
Yeah, it's a bit of jargony term.
but stick with us, folks. Essentially, we're talking about those yes or no situations you often see in research, like you either have a disease or you don't.
Right, or you're taking a certain medication or you're not, things like that.
Exactly. But here's the thing. A lot of those yes-no situations we study, they're actually really simplified versions of something that's way more complex in real life.
They are, yeah. It's like we're taking something that naturally exists on a spectrum and just chopping it in half.
Yeah, like that example from the paper we're looking at, hypertension.
Hmm.
We often just label someone as having hypertension or not based on a single blood pressure reading.
Right. And that can be misleading.
Totally. Blood pressure isn't an on or off switch.
Exactly. Blood pressure exists along this whole continuum. And by trying to force this continuous variable into a yes-no category, well, it can really distort our understanding of what's actually going on.
So instead of seeing the full picture, all those shades of gray, we're stuck with just black and white.
Exactly. And that's where things get really interesting when we bring Mendelian randomization into the mix.
So it's like we're seeing you're either tall or short based on just like some random height cutoff.
Yeah.
Without actually knowing how tall anyone really is, we lose all those shades of gray in between.
Exactly. And in Mendelian randomization, those shades of gray, those nuances, they really matter.
Right, because we're using genes to try to figure out if one thing causes another.
Right, exactly. And if we try to oversimplify things, just make it a yes-no, and there's really a whole spectrum there,
Yeah.
it can really throw off our results.
Okay, so walk me through this then. How does simplifying things into these binary categories actually mess with the whole Mendelian randomization process?
Okay, so let's stick with that blood pressure example. Let's say there's a gene that influences your blood pressure. It might only have a tiny effect, just a
Right.
little nudge upwards. Now, someone could have this gene, right? But maybe their blood pressure doesn't quite cross that line, that threshold we've set for saying, okay, this is what we're calling hypertension.
Because we've made it this binary thing.
Exactly. So we might look at that and think, oh, well, that gene doesn't really do anything important. But here's the thing. That slight increase in blood pressure, even if it doesn't push someone over that arbitrary cutoff,
Yeah.
it could still increase their risk of heart disease down the line.
Oh, wow. So even if they don't meet the official criteria for the exposure, because we've made it this binary thing, the gene is still having an impact.
Exactly.
Sneaky.
It is sneaky. And this really throws a wrench in the whole Mendelian randomization machinery.
How so?
Well, it violates one of the core assumptions we make when we use Minar racks, that the gene we're using is actually impacting the exposure we're interested in. We're trying to isolate the effect of hypertension as a cause, right?
Right.
But because we're missing all that nuance in the underlying blood pressure, because we've forced it into this yes-no category, we're not truly isolating that effect.
It's like we're trying to study the effects of a dimmer switch on a light, but we can only see if the light is on or off. We're totally missing all those levels of brightness in between.
That's a great analogy. And it brings us to another really important point that this paper highlights: the whole issue of compliers in Mendelian randomization studies.
Okay, compliers, haven't heard that term before. What's that all about?
So remember, one of the big assumptions we make in MESSER is that the gene we're looking at is actually influencing whether someone is exposed to the thing we're studying. So in this case, with our blood pressure example, the gene would need to actually cause a big enough difference in blood pressure to actually push someone over that line and be diagnosed with hypertension.
So they're complying with what the gene is doing.
Exactly. The compliers are the people whose exposure status actually changes based on the genes they have.
Okay, that makes sense. So what's the problem then?
Well, the problem is this. With these binary exposures, especially when they're based on something that's really a continuous variable underneath, those compliers are likely a very, very small group.
Oh, so it's not representative of the whole population.
Exactly. Not even close, probably. And that means the results we get from that MR study,
Yeah.
they might only apply to this tiny specific group of people whose genes have a very particular and strong effect.
So it's like doing a drug trial for a new weight loss medication, but you only include people who are already like incredibly sensitive to changes in their diet and exercise.
Exactly.
It wouldn't tell you much about how the drug would work for the average person.
Exactly. The results might not be generalizable at all.
That's a really important thing to keep in mind when you're reading these studies.
Absolutely. Especially when you're dealing with binary exposures like this.
So we've got these two layers of complexity then, forcing these continuous variables into these binary boxes, and then the whole issue of compliers potentially skewing the results. It's almost enough to make you want to throw your hands up and say, forget it, studying this stuff is impossible.
Well, it definitely makes things tricky. But that's part of what makes this kind of research so important. And this is where this paper brings up a really crucial point about power calculations.
Okay, power calculations, taking me back to Stats 101 here. That's all about figuring out how many people you need to include in your study to actually be able to detect a real effect, right?
Exactly. And the paper makes the point that with these binary exposures, especially when they're coming from an underlying continuous variable, the power calculations often end up being way too optimistic.
Anyone.
Meaning researchers might think they have enough data to find a real effect when they really don't, because they've oversimplified things, right? Made it a yes-no when it's not. They're not fully capturing all that nuance in the data.
That's not that the studies are intentionally misleading or anything.
Right, exactly.
It's more like the way we're asking the question is actually preventing us from getting the full answer.
That's a great way to put it, yeah. And it raises a really interesting question, I think. How many potential discoveries are we missing out on? How many clues about what really causes or prevents disease are we just completely overlooking? Because we're not looking at the data in the right way.
Kind of a humbling thought.
It is, isn't it? It's like, even with all our fancy scientific tools and techniques, sometimes the biggest roadblock to discovery is our own way of thinking about the problem.
Absolutely. So it sounds like the big takeaway here, for anyone who's reading these studies, whether it's for their work or just out of curiosity, is to approach those headlines, the ones that are like X causes Y, with a healthy dose of skepticism, especially when it involves one of these yes-no exposures.
I think that's great advice.
Don't be afraid to think critically about it. Question the assumptions that are being made. And maybe even take a minute to think about that underlying continuous variable that's hiding beneath the surface.
Exactly. Don't just take the binary at face value. Think about what else might be going on.
Well said. This has been a fascinating deep dive.
It really has.
Thanks for joining us, everyone. And remember, keep those brains buzzing and those questions coming. We'll see you next time.